All right, so a little history about how genetics were thought to be. In the past, there were kind of two ideas about how we thought genetics worked. The first one was this blended theory right here. If dad is tall and mom is short, then the offspring are going to be kind of medium height, right? If you end up having a golden retriever and a poodle, you end up getting a golden doodle, right? If you get a bulldog and a shih tzu, right, you can imagine what you get. A bull shih tzu, right? So there's this idea that if you have dark-skinned and light-skinned parents, you're going to get this medium blend, right? So there was this blended theory that's how it worked with all traits. But then we saw sometimes that traits, they're not blended, they completely disappear, right? If you have a tall plant with a short plant, you end up having like no all tall plants. So sometimes they develop what we call the particulate theory, which is traits are inherited as particles. You get all these little particles from dad and particles from mom, and sometimes dad particles cover up mom's particles. And so what Mendel did was he kind of clarified those two previous theories. And so Mendel is considered the father of genetics. Well, first of all, what is genetics? The word gene wasn't even identified at the time of Mendel. This was something that we developed afterwards. The study of genetics is the study of heredity. And heredity is where you take from one generation, right, your parents, and what we pass on from the next generation. Here's what we call F1s. We'll learn later that F stands for filial, which means like filu or fila, which means son. This son right here, not this son, right? And then the kids pass on their traits to what we call the F2s, and then those kids pass on their traits to what we call the F3s. So heredity is how we inherit. Inherit, that's where we get the word heredity. If I get a tattoo, are my kids going to be born with my tattoo? No, that would be weird, right? Those are traits that are acquired, right? If I'm in a car accident and I lose my left arm, my children aren't going to be born missing an arm, right? So that would be weird. Those are not inherited traits that you're born with. And we learn now that our inherited traits are controlled by what we now know to be genes, which are little pieces of DNA that were given to us from the sperm and egg cells of our parents. So Mendel used peas. He studied all this by looking at pea plants. But one of the reasons why he used pea plants is that they grow fast, right? And they have very distinct traits that have what I like to call this either or outcomes or versions. They grow fast, so within a couple of weeks you can go from one generation to the next generation to the next generation, so you don't have to wait years and years and years to see what would happen if you crossed a tall plant with a short plant, what would happen if you crossed a plant that had purple flowers with a plant that had white flowers. And so he looked at like 7 different traits, for the most part, 7 main ones. He looked at the pod shape, he looked at the seed shape, he looked at the seed color, he looked at the pod color, he looked at the flower color, he looked at the plant height, he looked at the flower position, whether it came on the end or the sides of the stems. He looked at like 7 main traits. And the reason why he chose those 7 traits is because when he looked at those traits, there was 2 versions. Either tall or short for height. This is for height. So this is the trait, right? And these are the versions. What do we call the 2 versions of a trait controlled by a gene? It starts with an A. These are the 2 alleles. Right, so you have the tall allele and the short allele. When it comes to the trait, so the trait here would be flower color. Right? And this would be seed shape. Either kind of round or wrinkled, purple or flower. Now we don't know which is dominant and which is recessive yet because we haven't done any experiments. And so in order to understand how Mendel did these experiments, you kind of have to understand how plants have sex with each other. And the sex organs of a plant are the flowers. If you look at a flower, all flowers have a basic structure. They have these things called stamens, right? That on the tips of them are these anthers that have little kind of, that's where the pollen is. Think of the pollen like the sperm from animals. Think of the pollen as like the sperm of a plant. Pollen is created by the process of meiosis. So these cells are the sex cells or the gametes. So what do we know that's different about sex cells and gametes than every other cell that makes up this plant? They're haploid. Thank you, Darren. They're haploid. So the pollen is haploid, meaning it has half of the DNA. So remember, our chromosomes comes in pairs. This is dad's, this is mom's, right? Remember homologous pairs, we line them up. Well, actually, they were like this, remember? And then we separate them. So half of the DNA, right? In humans, you'd have 46 chromosomes. 23 of the chromosomes would go in this, and 23 would go in that. So the plant, here's the pollen, and right in the middle of the pistil are the ovaries, or the eggs. So there's your egg, and there's kind of like your sperm. Think of it like that. A plant can have sex with itself, okay? In the sense that it can reproduce on its own. It's like if you had a penis and a vagina and you could give birth to your own baby. Stop letting those thoughts go too far before you'll let your mind wander in a dark, dark place you can never come back from. But the way that this happened is, let's say I have a little buzzy bee. This is my bee. Look at my bee. Bzz, bzz, right? That's my stinger. You know what? That's all I got for you right now, okay? This guy lands in here, and he gets kind of, he's looking for some nectar, and some of the pollen gets sprinkled, it kind of moves onto the tip of the pistil. It works its way down and fuses with the egg. So you have, this is a sperm or the pollen, half of the DNA, it fuses with this, and then now you end up having a zygote, right? A fertilized egg. This is the seed, okay? So what ends up happening is all of these things kind of fall off, and this thing grows into the pea pod. So you got one pea, and then the peas kind of duplicate themselves, and all of those get planted, and then they grow into the plant, right? This is my plant. You guys like my plant? Here's the flower. Isn't that beautiful? Come on, give me some props. Right? And so so we can self-pollinate, but that's what self-pollination or self-fertilization looks like. What about cross-fertilization? What if I got this tall plant and I wanted to fertilize it with this short plant? So Mendel was the genius. He figured out a way to make sure that only the pollen from this plant, right, got into this flower and not its own. He took scissors and he cut off the male parts of the flowers, right? So he trimmed these off. It's a flower. Relax, guys. Nobody's getting castrated. And so then he took like a paintbrush. He took like a paintbrush, right? And he kind of brushed the pollen onto the paintbrush and then went to the other flowers and flicked off the pollen directly onto that flower. So it was he who controlled the breeding. And so he could now cross-breed or cross-fertilize. Got it? Yeah, hold on one second. So he wanted to start working with what we call true breeding or pure breeding plants. These are plants that always create plants that look like themselves. So what he did was he would take a tall plant that every time it fertilized itself, it produced tall plants. And so if you took a tall plant and self-fertilized it, so if you go back to here, if this was tall and it created a bunch of seeds, all of the seeds produced tall plants. That's a true bred tall. And so what we know about that is both of its chromosomes are probably tall. Or are tall. And so no matter how you combine these two T's, you're always going to get a tall. Now, the short plants were also purebred. When they self-fertilized themselves, you got nothing but short plants. So what we have is, if he took a tall plant and he bred it with the short plant, and he ended up getting what we call hybrids, which are offspring of true bred or purebred plants. And so a hybrid is going to have a tall allele from the father. So the chromosome that was received from the dad had the tall version of that gene. And the chromosome that they got from the mom, whether it was from the pollen or the egg, had the short allele or version of that gene. And then Mendel grew the seed and wanted to see, is this plant going to be tall or is it going to be short? And guess what he found out? It was tall. And so from that, he determined that some alleles are dominant and some alleles are recessive. So the dominant trait kind of covers up the recessive trait. And when he grew the tall and short, he noticed that all were tall. So the very first generation we called the parent generation or P generation, right? They were a tall one and a short one. And the F1s, F standing for filial, which means son. This is the F1 generation. They were all tall. Okay? And so tall, obviously recessive. And if you do the Punnett square, it looks like this. And we'll get to Punnett squares later. You put one parent up top. Let's do the tall one, right? Big T, big T. And the short one, little t, little t. When you combine them, big T right here, little t right here, guess what? Big T, little t, that's going to be tall. Big T, little t, big T, little t, big T, little t. Because the big T is dominant over the little t. And that's what you get. What do we call the letters? Big T, little t. Does anybody remember? Starts with a G. No. That's the genotype. The gamete is actually these guys. This is the gamete, right? These would be like the sperm, and these would be like the eggs, or vice versa. Tall, which is what the genotype codes for, is the phenotype. And we'll get to those vocabulary terms again. We'll revisit those. So what we're inheriting are genes, but Mendel didn't call them genes at that time. The word genes was something that we didn't really understand because we didn't have the technology to look inside of the cells and see the DNA. But what is a gene? A gene is a piece of DNA. So here's a chromosome, right? This is a piece of the chromosome. Here's a piece. Let's unravel it and look at the DNA. From here to here is one little piece. That's a gene, which is instructions to make a protein, and that protein controls the trait. So that could be like flower color. Further on down the DNA, there's another piece of DNA on the same chromosome or another chromosome. Gene 2, which is a piece of DNA that codes for a protein. It's instructions that tells the cell how to make a protein, and that protein is in charge of or has a function that controls some other trait. So that could be like flower color or plant height. So genes, where are they located? They're located on what? And we have 23 chromosomes. Some chromosomes have over 1,000 genes, right? Fruit flies have like 4 chromosomes. We have 23 chromosomes. Chimpanzees have 24 chromosomes, right? Some ferns and plants have thousands of chromosomes. It's just how many pieces we break up our DNA into. That's all. All right. So we'll stop here. I'll pause it. All right. So Ethan, what is the father of genetics?
Gregor Mendel.
All right.
I'm number 2. I'm number 2.
Princess, genetics is a study of heredity. When you inherit something, that is heredity, which is how traits are passed from blank to blank. You are right. Parents to offspring. I had another class say generation to generation. That would also make sense, right? That would also make sense. F1 to F2, that's another way. That's good. F1 to F2 or P generation to F1 generation. Good, I like that. All of those are correct. Mendel studied what organism? Anna. Yep, you got it. Pea plants. This is just warm-ups, guys. It's pretty easy. Juan, if one trait covers up another, we say that trait is what? The one that's covered up, what do we call that one? Yeah. a true breeding, it's also, it depends on the text that you're reading and the worksheet that you have. I don't want you guys to confuse true breeding and purebred as anything different. It's the same thing. Yeah, those words are equal to each other. They are synonymous with one another. Is a true breeding plant one that can only produce plants like itself? True or false? We'll go with Jake.
True.
Yeah, that's true, right? So if like you have a white rabbit, they can only make white rabbits, things like that. If a tall and short plant are crossed, it will create a Stephanie. Yeah, correct. Hybrid, yeah, good. Right, you got a lion and a tiger, you get a liger. That's, are ligers real?
Yes.
Do they happen naturally in the wild? No.
Yeah, they have like a limited lifespan, but they also have a muscle.
They're not the same species. All right, so Mendel. So what he did was, he took his P generation, right? He took a tall plant, right? And by the way, the trait, which is controlled, the trait is height, right? The trait equals height. And so the big T is the dominant allele, which we call tall, and this is the short allele, right? Those are the alleles. Right? We've determined already, right, that tall is dominant over short, but the way that we've done that is by doing an experiment. So let me get rid of these guys real quick. So P generation, he did not know that yet until he did this. Oops, sorry. Tall and short, and then the F1 generation, he got all what, everybody? Got all tall. And so we can assume that it was like this. The letters, these we refer to as the? And then this is the? Yeah, phenotype, which explains what the trait would look like. Yeah, you're right. And so these are all going to be big T, little t. That's the genotype. But then, guys, these two plants produce many seeds. So from the P generation, Mendel planted hundreds of seeds in that F1 generation. And so he crossed 2 of those F1 generation plants. And to produce his F2 generation, right? And so what we see now is that 75% of the plants, whether he planted 100, whether he planted 212, whether he planted 468, approximately 75% were tall. Approximately 25% were short. So if he planted 100 seeds, 75 would be tall, 25. But it's not like he got exactly 75 and 25. He might have gotten 76 and 24. He might have gotten 73 and 27, right? Oops. It was approximate. So one of the things that he concluded was that at some point, right, we inherit 2 things from our parents, big T, little t. Right? Big T, big T. We inherit one thing from our parents. But when we go to make our own offspring, we separate them again. And that is the law of segregation. Really, the principle of segregation explains this. Meiosis. That we produce sex cells and gametes that have half of the DNA. We got to separate what we got from mom and dad before we make our own children, because we can't just combine both with both parents, because then you have double the DNA you should have. Oops. So here's your tall. Here's your short. That's the P generation. You ended up getting these hybrids, right? Okay, that's the F1 generation. And so if you were to combine that sperm with that, or that chromosome in the sperm with that chromosome in the egg, you ended up getting like a big T, right? But what if you combined this with that one? Then you get a big T and a little t. But there's also equal chance that you get this chromosome in a pollen, and it combines with this chromosome in an egg. So you got another big T, right? This one right here. That one goes right there. And this little t. That's a little t. But what if the only other possible combination is going to be this little t and this little t? And you're going to get this right here, which will give you a short. All of these are going to be tall, and this one. So there is a 3 out of 4 chance, or possible combination, that you're going to get offspring that have that genotype. So there's a 75% chance that your kids will become tall. Not to say that every 4 plants that you plant, 3 will always be tall. This is just to show what the possibility, what is the probability, what is the chances, okay? And you should get, based on chance, 25% short. What's the chance with humans to get a son or a daughter? 50-50. Can you have the 2 parents have 5 daughters in a row? Is it possible? Is it probable? No, but it's possible. So a Punnett square doesn't tell you exactly what you're going to get. It tells you what you should get based on probability. So the way that we set this up is we use Punnett squares. And a Punnett square, right, you put the parents' genotype up at the top, right? That's the parents. And on the side. And then these are the parents or parental alleles, right? This would be like what's in, this is like what's in the gametes, right? So if you look at it, if this is a big G, little g plant, right, one of the gametes is going to have the big G, the other gametes is going to have the little g. Now this is the trait for what? Pod color. Obviously, big G is the dominant allele, which is green pods are dominant over, and we use lowercase, right? Yellow pod. And so these are the alleles. All right, that's the alleles. And so if one of the plants is a hybrid, big G, little g, so this cross looks like this. That's one parent crossed with another. That's what this X means. That means I'm going to crossbreed these. Okay? And so I set up this Punnett square. And so when this combines with this, you get the genotype, big G, the phenotype, green pod. The genotype, big G, little g, the phenotype, green pod. This is the genotype, that's the phenotype, that's the genotype, that's the phenotype. Now, what if I had that cross? So take a minute, set up the Punnett square, and show me what it would have looked like. Okay, so first of all, let's set up. Let's put this guy, let's put this parent on the side over here, right? Let's put this guy. So I got big G and a big G. And then let's put this parent on the top. So I'll put a big G and a little g here. And so when we combine, we got big G, big G. That's the genotype. What's the phenotype? I'll write it below. Green. We're talking about also big G, it's going to be? Different genotype, but still? Different genotype, but still? So we end up doing these genotypic ratios where you got 2 different genotypes, right? You got 50% of them, right? 2 out of 4, and then 50% are like this. But if you look at the phenotype, phenotypic ratios, look, they are all what? They're 100%. So just because, they're all green doesn't mean that they all have the same genotypes. We can also look with seed shape. Round is dominant over wrinkled. We can look at flower color, white and purple, right? Purple is dominant over white. We can look at the color of the seeds, the seeds themselves, yellow dominant over green, okay? And so you can create all these different Punnett squares by looking at that. So with here, they chose to use, for whatever reason, big Y as round and little y as wrinkled. Why they chose that, I have no idea. I would have used what letters? No, don't use R and W. Just use capital R for the dominant and lowercase r, right? So if you bred two heterozygous round pea plants that had rounded seeds, it would look like this, right? Two wrinkled would look like this. So you have to understand this concept of genotype and phenotype. Let's first talk about genotype. If you have the same alleles for both chromosomes, what do we call that genotype? The same. Homozygous. This is the genotype. But there, because it's the capital letter, we call it homozygous. If I'm a big T and a little t, I refer to this genotype as being... No, you don't need to say dominant or recessive because you have one of each. The word heterozygous explains it all. That's all you need to say. If I say heterozygous, I don't need to tell you dominant or recessive. You just put one of each. But if I say homozygous, I have to be specific. I have to give you a second descriptive term to explain, well, is it the same big T, homozygous? Or is it the same little t? Well, that's where I would say, if it's little t, homozygous what? Recessive. So these are the different genotypes, right? The phenotypes would be, what does it stand for? So in the case of flower color, right, this would be a genotype, big P, homozygous dominant. Right, this would be homozygous. I'll write it over here. Dominant. Okay. And big P stands for the phenotype of purple. So big P, little p, also purple. Big P, little p, also purple. Little p, that's going to give you the phenotype of being white. Okay? Breeds composition. All right. Let's go with Cameron. Number 1. Oh, Cameron Pugred. Yay! Let's go with Jose. Who is the father? By the way, I'm all cut up with Walking Dead. Oh my God. And then I had to wait till February whatever. Oh my God, what? I have to wait for the next episode? Oh my God. God, what is going on? Alright, let's go. Princess, genes are located on? You are correct. Some people would say DNA there, and they'd also be correct. They are located on our DNA. Edwin, what's number 4? Each gene is made up of 2? You are correct. You get 2 versions of each gene, right, on each chromosome, one from mom, one from dad. Number 5, Silvano. You are correct. If you're looking at the outward appearance, like whether it's wrinkled or round, that's the phenotype. If it's big R, little r, big R, or little r, that's the genotype. All right, let's start up here. Let's go with Atticus. What's number 6? The letters RR. What do we call those? Correct. An organism that has 2 different alleles. Alondra. What do we call when they're different, like big R, little r? B, hetero? Yeah, because hetero means different. Homo means the same. An organism that has 2 the same. Class, what is it? Homozygous. Number 9, Mendel's parent generation, the very first one that he did. Edwin, what was it? Which one of these would best represent that P generation, that where he bred 2 purebreds? Huh? Nope. He didn't cross two that were the same. Yeah, in the back, Silvano, right? Which one is it? C, correct, right? He did one purebred and then another purebred for the other trait. Right? It would be like tall and short, purple and white, green and yellow, whatever. Lastly, let's go with Darren. When two different alleles occur together, such as big R, little r, what do we call that? And one is expressed. Only one is expressed. We call that allele the what allele? Huh? Dominant. That's right. So if you end up having one or the other. Okay, so what is the diagram shown below called? And what does this letter actually represent? And I want to add another question. What do the letters inside the square represent? So I'm talking about these, right? All right. Alani, what is this diagram called? Named after the guy who actually took Mendel's ideas and organized them. He was a mathematician into these squares. This is called Punnett squares. We're going to go with Dakota. What does this letter represent right here? This one. Yeah. Let's break it down into steps. Is it coming from the parent? Does that letter belong to the parent or the offspring? That's the parent? Okay. Is it the parent's genotype or just one of the alleles? Just one. This is one letter right here. Yeah. Okay, good. Now, this is, anybody want to add to that? It is recessive. It is it's one in the gametes. Yeah, absolutely good. You guys are all right. This is the parent. The parent will go through meiosis here. Meiosis, and he separates. This is Mendel's law of segregation, right? When we create our gametes, sperm or egg. So each one of these letters represents the parental gamete, sex cell, which should only have one allele in it. So then let's take a step further. Let's go with Cameron. Cameron, what do these letters inside of the box represent? Are they the parents or the offspring? Offspring. These are the possible offspring what? Somebody said it. Offspring genotypes. One eye is crossed with two eyes, and they're all two eyes. So then our trait, right? Our trait we'll say is number of eyes. And let's say that we have to pick a letter. I'm just going to, oops, what the heck is that? I'm going to pick E, right? So the capital E is the allele that represents what version of that trait? So what does the big E stand for? And what does the little e stand for? Okay? So if you use the letter E for this, what is the genotype of the offspring if the parents were this? If you were to do this cross, I'll just show you the cross. One of the parents, I don't care if you put it on the top or the side, is going to be this. The other parent, right here, if you cross them, if this gamete were to combine with this gamete, you get this offspring genotype. It's going to be the same around. So what's the possible offspring genotype? What do we call an offspring that is a combination of two different purebreds? That's a hybrid. If you cross the offspring with each other, how many new offspring would you expect? So now you're going to do this. Okay? So take a minute to do. I want you guys to check your answer and see if this is what you got. I'm going to do a little mini Punnett square right here. We're going to do two hybrids. You're going to have one that's a two-eyed with another that's a two-eyed, right? This guy's going to be two eyes. This guy's going to be two eyes. That's the phenotype. This guy's going to be two eyes. But this guy will be one eye. So out of the 4 possibilities, 3 will be
Two-eyed.
two-eyed. What that means is that 75% of however many children these monsters have should have 2 eyes, and 1 out of 4 should have 1 eye. So 25% should have.